Now it's okay. Okay, okay. So I am audible now. My voice is clear to all of you. Is my voice is clear to all of you? Now it's proper. Okay, good, 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 good. It's okay. So one more query. I will show you the PPT. You have to tell me whether we are here. We are on this PPT. We are on this PPT, or we have done it, or we are on this PPT. Whether we have done this PPT or not. Just let me know once a time, please. Please. फर्स्ट पीपीटी. ओके ओके. थैंक यू ऑल. थैंक यू. तो मैं अकेली ही बोलेगी इतना बोल दिया मैंने. मैम वही वर ऑन लास्ट स्लाइड दैट द वट एस डी एस वन. ओके ओके ओके. So okay, I will speak once again. I will start once again. We will start from here only. Okay, okay. So students will join by themselves only. They will get the link. They will get the link, na. So they will join once again. Okay. So okay, I am going to the PPT then. Yes, we are here. So this is sodium dodecyl sulfate polyacrylamide gel electrophoresis. In this electrophoresis, the main point is that the protein purification is done on the basis of size. This is the first point, that the protein separation or the protein purification has been done on the basis of their molecular size. So the molecular mass is quite important in this case. Then what is SDS? I told you that it is a negatively charged detergent. Basically what SDS is? SDS is negatively charged detergent which has two functions. So you have a protein molecule which is tertiary in structure. So as you can see, this protein molecule is tertiary in structure. So this protein molecule may be either positive or negative or may be neutral in nature. So it may be of any type, like positive, negative or neutral. But when we are treating it with SDS, you can see here SDS. SDS is negatively charged. What the SDS will do? It has to do two things. Number one, this negatively charged detergent or this negatively charged SDS will make this folded protein into a linear one. And you can see it becomes a linear one. So protein is folded here like this one and it becomes linear one. Number one function. Number two, this protein molecule may carry any charge, positive, negative, any charge. The SDS will make this protein fully negatively charged. So the protein will become fully negatively charged. Let me know whether you are here. Let me know. So yes, I am starting. I am just doing it again, Amita. So you are getting me now. Are you getting me? Are you getting me now? Yes, ma'am. Okay, okay, okay. Good, 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 good. Okay. So yes, they are getting it. Okay, good. So this is the main point which I want to discuss, that the SDS has two functions. This SDS has two functions. Number one function is to linearize the protein, to make the protein linear in structure. This is one function. And number two is to make the protein negatively charged. To make the protein negatively charged. So when I will ask you this question tomorrow, you must know it, that it has two functions, to make it linearized and another to make it negatively charged. So SDS dodecyl sulfate polyacrylamide gel electrophoresis is a technique which is widely used in biochemistry, forensic, genetics, and molecular biology to separate and identify the proteins according to their molecular weight. You have to keep this point in mind only. So, we will separate the proteins, or we will distinguish them according to their molecular weight. So, this method separates protein based primarily on their molecular weight. So, molecular weight is the main point here. Now, we have to go with the principle. I will try my best to, so that you can understand everything very nicely, because SDS page is very much important. So, let's start with the principle. SDS is a detergent which is negatively charged, we know, which denature the proteins, okay, by binding to hydrophobic regions. All non-covalent bonds will disrupt and the protein require a negative net charge. So when we put the SDS solution, or when we put SDS, what will happen? The protein becomes linearized and becomes negatively charged. First point. This is the first point. Number 2. Then we have another thing which is beta mercaptoethanol. Beta mercaptoethanol. This is the second point. A concurrent treatment with disulfide reducing agents such as beta mercaptoethanol or DTT, also known as dithiothiritol, which further denatures the protein by reducing disulfide linkage and thus overcoming some forms of tertiary protein folding and breaking up the quaternary protein structure. So basically beta mercaptoethanol or DTT is also using for the same purpose to denature the protein, because protein is found in primary structure, secondary structure, tertiary structure and quaternary structure. So to make it simpler one, to make it linearized, we have to use beta mercaptoethanol. So first we are using SDS and now we are using beta mercaptoethanol. For what purpose? To break the quaternary structure, to break the protein's quaternary structure. So the protein samples are having uniform structure and charge. The separation will depend upon their molecular weight only. So what we will get, बच्चे हमें क्या मिलेगा? हमें वो प्रोटीन मिल जाएगा. We will get that protein which have a uniform or a linearized structure. So this is the protein which we get with negative charge all over. With negative charge all over with the help of this SDS. And now this protein will be separated in the electrophoresis. Are you getting my points? So electrophoresis is as same as we have discussed before also, that smaller proteins migrate faster through the gel under the influence of applied electric field. Obviously the proteins which are smaller in size, which are light in weight, they will move much faster as compared to the larger one. So the number of SDS molecules that bind is proportional to the size of protein. Thus, in the electric field, protein molecule move towards the anode. So, why they will move towards the positive end? Suppose this is our gel. Let me draw here. This is our gel. This is the negative end, and this is the positive end. So, from in which end they will move? They will move in this end. Why they will move in this end? Because they are negatively charged, so they have to move towards the positively charged end. Okay, they are negatively charged, so they will move towards the anode or towards the positive end and separated according to their molecular weight. Now this is the diagram, not a diagram, this is just a presentation basically. We have a protein. You can see the protein is what in shape? Highly folded, highly quaternary or like a tertiary or a quaternary structure. So by the treatment with SDS, what will happen? It will become linearized. You can see here, it will become linearized. And as SDS is negatively charged, this one is SDS. This one is SDS, negatively charged. As SDS is negatively charged, it makes the protein negatively charged, and this negatively charged protein will move towards the positively charged end. So this is the one case. Now we also use mercaptoethanol. We also use beta mercaptoethanol. What's the function of this one? Beta mercaptoethanol is generally used to break these disulfide bonds so that the quaternary structure will become simplified. Okay. Let me check whether you are getting it or not. Kindly reply me. Yes. Are you getting it? I am just waiting 1 minute, please. So are you getting it, students? Kindly let me know. Are you getting it? Yes, Priya is here, Gayatri is here, Siddhi is here, Rahul is here, Amita is here also. She is getting it. Very good. So we have two things till now. We have SDS, which is a detergent which is negatively charged, which make the protein linear and negatively charged. Okay, first point. Then we have mercaptoethanol, which breaks the disulfide bands. The disulfide bands will be break and the quaternary structure will become the linearized structure. So we have done the 2 points till now. So is it clear to all of you or not? Kindly let me know. Sakshi is also here, Sejal is here. Well, you are kindly reply, all of you please reply. Rahul is saying yes ma'am, Rahul is getting it. Where is Jeet? Where is Khan Maria? Manpreet, kindly reply me. Manpreet, where are you? Are you getting my point? Juveria, where are you? Kindly reply, बच्चे. Vaishnavi is here, Kaksha is here, okay, Pooja is here, okay. Chalo, let's move then. You are getting it, okay, good. So, this is the 2 points which we have discussed till now. Now another thing is, why we are using page? Why we are using polyacrylamide gel? Why we can't use agarose? So in the very first day I have asked you that tell me the difference between page and agarose and agar. So why we are using page here? Why not we pursue it on agarose gel electrophoresis? It is also electrophoresis. It is also for the separation of biomolecules. Why we are using page? So there are some points which are important. Number 1. The size of the pores is determined by the concentration of acrylamide. So, the pore size, the size of the pores on which the protein has to run is very important. The size of these pores will basically depend upon the concentration of acrylamide. Like we have a agarose powder, we have a acrylamide. The higher the concentration, please remember this point or write it down. The higher the concentration, the smaller the size of the pores. So if you are using, let me explain, like if you are using 0.3% or you are using 0.9%. So in 0.3 or in 0.9%, in which percent the size of the pores will be small? Answer me it first, then I will move further. Higher the concentration, smaller the size of the pores. So you have 0.3% polyacrylamide, you have 0.9% polyacrylamide. In both of it, in which the size of the pores will be small? Kindly answer me. In which the size of the pores will be small? Kindly let me know. Kindly let me know. Yes, yes, come on, reply, come on, good, good. Yes, yes, come on. Yes, Prachi is here, Jagruti is here. Yes, Gayatri and Manpreet is here, Vaishnavi is here. Very good, very good. So, more the concentration of the gel, smaller the size of the pores will be. जितना हम ज़्यादा concentration लेंगे, the size of the pores will get smaller and smaller because it becomes more authentic for the separation of proteins. So, more the percentage of the gel or more the quantity of the gel, the smaller the size of the pores. Okay, you are getting it. Okay, very good, very good. Mitali is here, very good. Siddhi, Anushri, Sejal, very good बच्चे. Hetal, Pooja, Rahul and Sakshi, very good to all of you. So let's move now further. So I have 2 points which I have to discuss. That higher the concentration, smaller the size of the pores. So write it down anywhere that जितनी हम ज़्यादा concentration में acrylamide लेंगे, then the smaller the size of the pores. So we have to study STS page which consists of two different gels. So in STS page which we are using these days, we have two gels. We have stacking gel and we have separating gel. So in the above portion we have stacking gel and in the lower portion we have a separating gel. Like agarose is a continuous gel. You put the samples in the walls. You put the samples and then you have to run it. But here we have two concentrations of the gel. So if you can see in stacking, the first one we have 4% of acrylamide and in the second one, the separating, we have 5 to 15% of acrylamide. So in which case the size of pores will be small? Either in stacking or in separating, in which gel the size of pores will be more authentic? In which case? Either in stacking or in separating. Kindly reply. You have 4% gel, 4% acrylamide and you have 15% acrylamide. So in which case the size of the pores will be small? Kindly let me check your answers. Yes, yes, come on, very good, come on. Write down बच्चे. Yes, very good. So, the lower one, the separating gel which is the lower one, it is more authentic or it needs more time for the separation of proteins as compared to the above one which is the stacking gel. So separating gel, that's why its name is separating, because it will take its time and it will separate the proteins according to their molecular weight and very slowly because the size of the pores is very small. It will take time as these techniques are time consuming, we all know it, but the separation is very perfect in this case. Yes, very good. So the separation in this case is very perfect in case of separating gel. That is why it's known as separating gel, because it helps in the proper separation of proteins according to their molecular weights. So this is the important point you have to remember. So which gel is more authentic or which gel is more good for separation? 15% of acrylamide, or also known as separating gel. That's why its name is separating, because it separates the proteins. So my question is that why we are using page? Why we are not using agarose? Because the number one answer is page is more authentic in the results. Because of its pore size, it gives the best results for protein detection, because proteins have little bit higher molecular weight, or you can say they have protein has primary, secondary, tertiary, quaternary structures. So polyacrylamide gel is perfect for the protein separation. So another points are that they are chemically inert. They will not react with the protein molecules. They are electrically neutral. They not carry any type of charge. They are hydrophilic in nature, and they are transparent for optical detection, that you can see it under UV light. This case is also same in case of your electrophoresis, agarose gel electrophoresis also. So this is the main thing, students, which I am talking to. How to prepare the gel, which is basically page. So we are discussing the page. So here this is the gel apparatus. You can see this is the resolving gel, or you can say the separating gel. So this gel is in the lower one is a separating gel, and this one which is very close to the combs. So these are the combs, like these are the combs where we have to put our sample with the help of pipette. So up to this there is a gel which is known as stocking gel, and after it the lower one, this one is known as the separating gel. So we have this gel in which stacking gel is the above one, and separating, where we have to separate the proteins, separating gel is the lower one. So now discuss the process of SDS paste. Like electrophoresis, we use agarose, and we boiled it for 10 minutes, and then we put it in a apparatus and then allow to cool it down. Similarly here it is same again, but little bit difference is there also. So first we will do boil the samples for 10 minutes. Why we are boiling it? To denature the protein. Protein is in complex structure, we have to make it linear structure with the help of SDS. So when we are boiling the sample, we must have SDS in it. Then we have to put this protein molecule into the gel apparatus. Then we have to pour the buffer solution, we have to put the molecule, or we have to put the sample, and then we have to run the electrophoresis chamber. Now you can see here, these are the SDS coated proteins. This is the first step. So where we are saying that proteins are denatures the proteins, we have to boil it for 10 minutes. So in this tube we have proteins. Protein is heated now, it becomes linear now, and it becomes negatively charged now. So in here the protein is linearized, and obviously it is negatively charged. Then this is a comb where we have to put it. Now the protein will migrate. If you can see the protein, these are different the size of the proteins are different here, like some are larger one like this one, and some are smaller one. So when we do the electrophoresis, or when we run the SDS page, what will happen? The smaller proteins, like this is the smaller protein, this will move first, because it is small in size, it will move first. Then we have the medium proteins, and then we have the large proteins. So the proteins will move according to their size. Smaller proteins will move first, after it we have medium sized proteins, and after it we have large sized proteins. So this is the thing they are mentioning, that SDS proteins, this is SDS treated proteins, have similar charge to mass ratio and similar shapes during page. So this is page, this is polyacrylamide electrophoresis. The migration of these proteins will depend upon their molecular weight. So everything is depend here on the molecular weight. Smaller the proteins, they will migrate faster through the gel, they will migrate fast under the influence of applied electric field, whereas larger proteins, the heavy ones like this one, this is heavy one, this is heavy one, so they will move slowly, they will move little bit slowly. So is it clear to you? I hope it will be clear to you. So this is again that SDS is an anionic detergent, negatively charged. The sample is first boiled for 5 minutes in the buffer containing beta-mercaptoethanol and SDS. So we have to take the tube in which we will put our protein molecule along with beta-mercaptoethanol and SDS. So what beta-mercaptoethanol, what will it do? Break the disulfide bonds. And what SDS will do? Make it negatively charged and also denatures it. So this is the thing we have done before. So you can see here before SDS the structure is highly complex in nature. Bache, it's highly complex in nature. But after treatment with SDS it becomes linear along with negative charge. So now this is again the thing which we are talking about. We have stacking gel and we have separating gel, also known as running gel. So stacking gel is just below the combs. You can see here the combs, the wells where we have to put the sample. We have to put the sample here. Like they are also saying, load the samples here. So we have to put the samples here. And this is negative side and the proteins will move towards the positive side. Why they are moving toward the positive side? Because they are already negative here. So smaller proteins will run faster and the bigger one remain on the upper side. So this is the you can say vertical electrophoresis. Like in gel electrophoresis, agarose we will put it down like that. We will put agarose in the horizontal position. But in this case, in protein separation, we will put the gel in the vertical position, so also known as vertical electrophoresis. So is it clear up to now? Let me check please. You are getting my point? You are getting my point, students? Yes, yes. Come on, come on. Good, good. Understand it, please first. Okay, okay. Kindly reply so that we will continue. Come on. Reply, students. Please reply. Yes, we have Sakshi. We have Pooja, Siddhi, Rahul is here. Aditi is also here. Juveria is here. Priya is here. So, okay. So you are getting it. So let's continue then. Okay. So this is the apparatus of the, you can say, vertical electrophoresis or SDS or Western blotting, or for the process of protein separation. So we have two gels, stacking gel and separating gel, also known as running gel. It's okay. So in the stacking gel, we have 4% of acrylamide, we already discussed before, and in case of running gel, we have 15% of acrylamide. So this gel, running gel, or you can say separating gel, this is the actual zone of separation of protein or molecules based on their mobility. So this is the actual zone of protein separation. The lower one, this one, if you can see here, this one, this is the actual layer of protein separation. So let me start this diagram. We have wells, like this is the wells. Up to this means this one. This one is the stacking gel, this one, and the lower one is the separating gel. So we have two gels, stacking gel and separating gel, and there is the valve where we have to put our sample. So kindly focus here. It's negatively charged, it's positively charged. So the ions or the molecules will move towards this direction according to their negative to positive attraction. So what happened here? Now we have to understand the pH here. Here the pH is 8.3, really slightly alkaline in nature. Here. Then we put our samples. We put our samples in the concentration of 6.7. We put our samples in the pH of 6.7. And the stocking gel also has a concentration of 6.7. But when we move into the separating gel, the pH increases. First the concentration is 6.7, but later on the concentration becomes 8.9. So what will happen? We will see here. We have, when we are using the buffer or when we are using the SDS or the chemicals, we have three things here. One is glycinate. We have glycinate, we have chloride ions, and we have a protein molecules. Chloride ion is negatively charged, glycinate is negatively charged, protein is negatively charged. All three are negatively charged. But we have to know which one will come fast, which one will come first. What happened? in case of this, when the pH is 6.7, here you can see the molecules are mixed. Like we have red dots, we have green, we have oranges, we have this porous gel is page, and these long threads are what? Basically this is the page, and these are the molecules which we have to separate. These one, the dots one, the green, the red, the orange, these have to separate. So what happened? Basically they will move. First they have to move in the stocking gel, and then they have to move in the separating gel. What will happen? The chloride ions, they will move faster. The chloride ions move faster, and then we have the protein molecules, and then we have the glycinate. So in the stacking stage, protein molecules are in between chlorine and the glycinate molecules. Protein molecule is in between. That is why it is known as the stacking stage, because the proteins are stacked in between. But when we move in the separating channel, or when we are moving in the separating gel, what will happen? The pH will change from 6.7 to 8.9. What will happen? Like there they are showing, this is a stage in the stocking gel. First chlorine will move, then protein will move, then glycinate will move. So protein comes in between. So that's why we are saying it's a stacking stage, where the protein is in between the two. But later what will happen? If I have a diagram somewhere, like here, when we are come in the separating gel, what will happen? The pH increase. Here the pH is 6 point something, but here the pH is 8. So when our pH increases, what will happen? Proteins ion will come first. I will show you somewhere. I will show you in the video also. So proteins ions will come first. You have to remind this thing. So if you can see here, in the now in the separating gel, we have a separation. These molecules, and these molecules are separated. These threads are basically what? They representing the gel, that is page. So you have to remember one thing: in the stacking gel, chlorine will move first, then we have protein molecules, then we have glycinate. So protein is in between the two molecules, that is why it's known as stacking gel. This is again the apparatus. You can see this is the thing we have done, that the molecule becomes linear in shape. You know it. So in separating gel, the lower one, Proteins separate owing to molecular sieving properties. Smaller portions pass more easily and larger ones remain in friction. This is the generally the structure of page or the polyacrylamide gel. So this is the main thing. Here the pH is less, so the protein will remain in between the two. That means they are stacking in between two. But when the pH will increase, the protein molecules will separate faster. And this is the buffer which is used in case of SDS paste. We have 10% SDS, we have tris chloride. So you must ask me, ma'am, from where these chloride ions come. Like here I am saying the chloride ions, from where it comes? It comes in the buffer. So buffer is basically made up of tris HCl. Buffer, you understand what is buffer? The solution in which we put the proteins. So the chlorine ions come from it, and we also add glycerol or glycerine. So SDS is here for making it negatively charged. Chlorine is also here. Glycerine is also here. Beta mercaptoethanol is also here. Beta mercaptoethanol, what will it do? It will denature the proteins. And then we have bromophenol blue to give it a coloration effect. So this is a buffer. The very first step when we have to make the protein linearize. So in the acrylamide we have tris HCl, we have acrylamide, like we have agarose powder, we have acrylamide stock, we have water, we have SDS, we have ammonium, and we have tint. So this is the concentration of buffer. This is the concentration of separation gel, the lower one, and this is the concentration of stacking gel, the above one. The pH is 6.8. Here the pH is less, and you can see here the pH is 8.8. You have to remember the pH of stacking as well as separating solution. In the stacking solution the pH is less, approximately 6.8. In case of separating gel we have a pH which is 8.8. So first the pH is low and then the pH is high. So here the pH is low, and also acrylamide solution, water is here, SDS is here, pH is low. So you can see here we contain HCl, that means chloride, we have glycine, we have SDS. So this is the final product which we will get, students. This is the final product. The bands which we are getting, this is the final product. So you have to understand it. Now we will just revise it once again. So these are the proteins which we get isolates from the cell culture. These proteins are then we treat with our SDS, our beta mercaptoethanol. Now these proteins become negatively charged. As you can see, these become negatively charged. Now when we boil it for 10 minutes, they become linearized. Now this sample is ready for to put in the gel, and then they will move according to their size. So the smaller one will move faster as compared to the larger one. The smaller one will move faster as compared to the larger one. I hope you are getting it. Please let me know. Let me check. Are you getting it? Yes, I will tell you why it is vertical, not in horizontal position. I will tell you. But are you getting it? Are you getting my point? It is little bit more advanced as compared to agarose one, but it is very important and very useful technique. So you have to remember it. I will ask lots of questions from you tomorrow. Okay. Kindly reply me. Now we will see one video so that whole things will be more clear to you. Okay, I am sharing one video with you. Okay. So this is only a virtual presentation, so it will be useful to you. That's why I am showing it.
Method and very powerful technique in the study of proteins. In SDS page, proteins are separated in a polyacrylamide gel based on their molecular weight. For sample preparation, a loading buffer containing SDS, beta-mercaptoethanol, bromophenol blue, and glycerol is added to the protein samples. Protein samples may be biologically derived, for example from prokaryotic or eukaryotic cells, tissues, viruses, environmental samples, or purified proteins. When the sample buffer has been added, the samples are then heated at 95 degrees Celsius for 5 minutes. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain. Proteins are formed by linking amino acids with peptide bonds and fold into one or more specific spatial conformations, driven by a number of interactions such as hydrogen bonds, hydrophobic interaction, disulfide bonds, and ionic bonds. SDS is an anionic surfactant and contain a polar head group with a net negative charge at the end of a long hydrophobic carbon chain. SDS denatures the native proteins by disturbing the hydrogen bonds, hydrophobic and ionic interactions, while the reducing agent beta-mercaptoethanol is used to cleave the disulfide bonds. Also, when a protein mixture is heated, SDS binds uniformly to the protein, and the intrinsic charges of this protein become negligible when compared to the negative charges contributed by SDS. This treatment brings the folded proteins down to linear molecules with net negative charge. Therefore, these proteins can be separated in a polyacrylamide gel based on their molecular weight. For gel preparation, a separating gel solution with pH 8.8 and a stacking gel solution with pH 6.8 are used, and both consist of acrylamide, bisacrylamide, ammonium persulfate and T-med. The gel is produced by polymerization between two glass plates anchored vertically in a cassette. The separating gel is poured first. The polymerization reaction is a vinyl addition polymerization initiated by a free radical generating system. Ammonium persulfate is used as the free radical initiator, while T-med accelerates the rate of formation of free radicals from persulfate. These free radicals convert acrylamide monomers to free radicals which react with unactivated monomers to begin the polymerization chain reaction. The elongating polymer chains are randomly cross-linked by bisacrylamide, resulting in a gel with a characteristic porosity. The pore size of the gel is related to the ratio of acrylamide to bisacrylamide. Low concentration is preferred for high molecular weight samples. The stacking gel solution is poured on top of the solid separating gel, and a plastic comb placed on the top of the stacking gel during polymerization enables the formation of small wells in the gel. Once the gel completely polymerizes, the comb is removed, and the gel cassette is placed vertically between two electrodes. Positive electrode located at the bottom of the gel, whereas negative electrode positioned at the top of the gel. The gel is inserted into a chamber, and a tris glycine chloride buffer system with pH 8.3 is poured to allow the conduction of current through the gel. SDS is also present in the gel and in the running buffer to make sure that once the proteins are denatured, they stay that way throughout the run. For sample application, in addition to the samples, a molecular weight size marker is usually loaded onto the gel. This consists of proteins of known sizes and thereby allows the estimation of the sizes of the proteins in the actual samples, which migrate in parallel in different tracks of the gel. Each sample is pipetted into its own well in the gel. And as we have seen previously, bromophenol blue and glycerol are present in the samples. Bromophenol blue is a dye that is useful for visualizing the sample in the well, and glycerol increases the density of a sample and used to make the sample fall to the bottom of the sample well, rather than just flow out and mix with the running buffer. After the sample application procedure, an electric field is applied across the gel, causing the negatively charged proteins to migrate across the gel away from the negative electrode and towards the positive electrode. Also, small proteins migrate relatively easily through the mesh of the gel, while larger proteins are more likely to be retained and thereby migrate more slowly through the gel, The stacking gel is used to ensure that all of the proteins arrive at the separating gel at the same time. Because gel wells are around 1 centimeter deep, so in the absence of a stacking gel, proteins would all enter the separating gel at different times, resulting in very smeared bands. As we have seen previously, the running buffer consists of glycine and chloride ions. So, in the sample wells we will have proteins, chloride ions, and glycine. Proteins and chloride ions are negatively charged. Well, glycine can exist in 3 different charge states depending on the pH. The isoelectric point or pHi of glycine is equal 5.97. At this pH glycine carries neutral charge due to the gain and loss of protons. At a pH below its pHi, glycine carries a net positive charge. And at a pH above its pHi, it becomes negatively charged. The running buffer pH is equal 8.3, which above the pHi of glycine. So, at this pH glycine is negatively charged. Once the electric current is applied, proteins, chloride ions and glycine are forced to enter the stacking gel, where the pH is equal 6.8. At this pH glycine switches predominantly to the zwitterionic neutrally charged state. This loss of charge causes them to move very slowly in the electric field. The chloride ions, on the other hand, move much more quickly. Chloride ions migrate in front of the proteins as leading ions, and glycine molecules migrate behind the proteins as initial trailing ions. Consequently, proteins are trapped and concentrated into a narrow zone between the chloride ions and glycine. This procession carries on until it hits the separating gel, where the pH switches to 8.8. At this pH, the glycine molecules are mostly negatively charged and can migrate much faster than the proteins. Proteins are separated, so higher molecular weight proteins move more slowly through the porous acrylamide gel than lower molecular weight proteins. Due to the relatively small molecule size of bromophenol blue, it migrates faster than proteins. And by optical control of the migrating colored band, the electrophoresis can be stopped before the dye and before the samples have completely migrated through the gel and leave it. At the end of the electrophoretic separation, all proteins are sorted by size and can then be analyzed by other methods, for example protein staining methods and immunological detection, such as the Western blot technique. After removing the glass plates, the stacking gel is discarded and the separating gel can be stained. Kamasi blue is the most popular protein stain. It is an anionic dye, which non-specifically binds to proteins. The structure of Kamasi blue is predominantly non-polar, and it is usually used in methanolic solution acidified with acetic acid. Proteins in the gel are fixed by acetic acid and simultaneously stained. After staining, the proteins are detected as blue bands on a clear background. The molecular weight size markers in a separate lane in the gel can be used to determine the approximate molecular mass of unknown biomolecules by comparing the distance traveled relative to the marker.
Okay Google, झांसी वाली रानी गाना
A vertical videos. Edvotech instructional videos. Separating proteins using SDS polyacrylamide gel electrophoresis, also known as SDS page. For this experiment, you will need protein samples, tris glycine SDS electrophoresis buffer, a vertical polyacrylamide gel electrophoresis apparatus, a micropipette and gel loading tips, a power supply, and a precast polyacrylamide gel. Step 1. Boil the samples for 10 minutes to denature the proteins. Step 2. While the protein samples are boiling, place the precast polyacrylamide gel cassette into the electrophoresis unit. Follow the directions provided by the manufacturer to ensure that the gel cassette is properly oriented. Step 3. Add the 1x tris-glycine SDS electrophoresis buffer to the chamber. The sample wells and the gel cassette should be completely submerged. Rinse out the wells using a small transfer pipette or micropipette. Step 4. Carefully remove the protein samples from the boiling water bath. Collect the sample at the bottom of the tube by centrifuging or by gently tapping the tubes on the tabletop. Step 5. Load 20 microliters of the protein samples into the wells in consecutive order. Step 6. Place the safety cover on the electrophoresis unit. Connect the leads to the power supply. Set the power supply to the required voltage and perform electrophoresis. Refer to the experimental instructions for time and voltage guidelines. Step 7. Electrophoresis is complete when the dye front has run to the bottom of the gel. Turn off the power supply and remove the gel cassette from the electrophoresis unit. Place the gel cassette flat on the lab bench with the large plate facing down. Step 8. Using a spatula, carefully separate the two plates of the gel cassette. Place the spatula in the slot at the top edge near the sample wells. Twist the spatula to separate the two plates of the cassette. Repeat until the plates are completely separated. Step 9. Gently lift the front plate away from the larger back plate. In most cases, the gel will stay on the back plate. If the gel partially sticks to the front plate, let it fall onto the back plate. Step 10. Proceed to staining the gel or to Western blot.
So student, this is basically the presentation. You can see the how the SDS technique generally works. So this is mainly that thing. So are you getting it? So it's not a difficult technique, it's a very easy technique, but you have to understand the pH change in the stacking and the separating gel. So in the stocking gel we have the pH which is little less, or it's in the separating gel we have a pH of 8.8. And one thing you also have to keep in mind that this is chlorine, yes, tris hydrochloride. So chlorine is negatively charged, it will move faster. And then we have the glycine, which have glycine is as an amino acid. So it may be acidic in nature or basic, or it may be neutral also. So first it is generally in the low pH, it is generally acidic in nature, that is it is carrying the positive charge. So chloride ions are there, then we have the protein, then we have the glycine. But when the pH get increased to 8.8, what will happen? The glycine will carry the negative charge and it will run very fast. So chlorine and glycine will run very fast and they will go down very easily. At that time we have only proteins left, which will take their time to get separated. So they will take their time to get separated. I hope you are getting my point, what I basically want to say. Yes, Amita is saying, ma'am, please send the video or PPT. Yes, we will share all these things. First let it complete today, then I will share it in the group. So you have to understand my point, what I want to say, that in the separating gel we have a pH which is approximately 8.8. At that pH, the lightweight negatively charged molecules like chlorine and glycine, they will move fast, so they will come first. and only proteins are left which will take their time according to the pore size, which will take their time to get separated. So kindly let me know whether you get it or not, then I will move further. Kindly let me know, please, kindly. Did you seen the videos? Have you seen the videos? Yes ma'am, yes ma'am. Okay, so they got my point which I want to ask. So we are here. So this type is the final result. So that in number 1 we have a ladder. So we know the molecular weight of these bands, like it is on what size, how many kilodaltons. So this one is a ladder, and then we have the samples. You can see the bands clearly. So this is the thing. Then we have to visualize it under the UV light. So when the bands are there, everything is there, then we have to check it under the UV light. Under the UV light we have to use the stain. Like we have two types of stain, like pumice blue and silver stain. Generally pumice blues are used basically. So we have to use to stain our gel. You can see here the gel is bluish in color. Are you getting my point? So it is treated with the pumice brilliant blue. So it is treated with a dye. So this gel is fully blue first, but after washing or after de-staining we will get the result. So we have to use this pumice blue dye to make the bands visible. So with the help of pumice blue we stain the gel. This is commonly used nowadays. So it's a traditional method followed by a de-staining. First we have to stain it. So in this state we still say stain the gel. So we have stained it with pumice blue, and then we have to de-stain it. What do you mean by de-stain? We have to wash it. So this is the types of stains which we are using. Now you can see the results. So you can see in the A we have done western blot, and in B we have used pumice blue, and in C we have used silver stain. So basically these are just the stains to give color to the bands. Otherwise how will we detect the bands if they are not showing any type of color? So these are the stains for coloring only. Okay, these are the stains which are used for coloring purposes. So what are the applications of SDS? To determine the purity of the protein sample. So we must know the purity of the protein samples, and the very important to determine the molecular weight of the protein, that how much weight it is carrying, to determine the how much kilodalton or how much weight the protein is carrying, and to identify the disulfide bonds. Then we have to quantify the proteins and blotting applications. These are the advantages and disadvantages of SDS. Basically this technique is quite commonly used because migration is proportional to the molecular weight. So smaller one will move faster and the larger one will remain in the larger one will move first, and the small molecules move first and the larger molecules will remain as such. Okay, but sometimes what happens, we get a poor bad resolution. So this is a negative point. They are highly sensitive test and separate 2% difference. So if we have 70 kilodalton protein, and we have 90 kilodalton protein, we can easily separate both of these proteins. So we can 2% difference of protein can be easily detected. But acrylamide which we are using is a neurotoxin chemical, so it should be avoided, and it requires small amount of samples and it provides us the accurate pore size. But one disadvantage is it is highly costly. So these are some of the advantages and disadvantages of the SDS. And what are their applications? Molecular weight का पता लगता है, peptide mapping, protein identification, determination of sample purity. Then we can identify the disulfide bonds. We can separate the proteins. This is another point. Blotting is there. Even the smaller fragments of DNA can be traced. So these are the applications. Advantages we already done. So this is the final results. The shining bands. We can use ethereum bromide, but basically it is used for DNA. We all know that ethereum bromide, like this gel, this is of ethereum bromide. You can see orangish bands. Are you able to see the orangish or the shining bands? This is because of this ethereum bromide. Then we can use comesty blue. Then we can use silver staining. So comesty blue is here, silver staining is here. So these all are the bands which we get in case of blotting techniques, or we can say in case of electrophoresis. These are again the applications. So this is the conclusion which I have to take. So SDS phase is a technique that used to separate the proteins according to their molecular size. Number 1. The separation is on the basis of molecular weight. The separation is on the basis of molecular size. Proteins are unfolded and migrate from cathode to anode. So first the proteins are highly coiled, they become single stranded, and they will move from negative end to the positive end. Second point. Molecular weight is determined by comparing the result with the standard curve of relative motility of the standard protein. So we will be able to determine the results with the motility of these standard proteins. So this is, I think, all about SDS phase, or you can say polyacrylamide gel electrophoresis. That's all for today. I hope you will get it. Are you getting it or not? Is the SDS or polyacrylamide gel electrophoresis is clear to you or not? Hello? Are you getting my point, students? Have you seen the PPT which I show you now? So in my live, I am just here on the YouTube video, but you have seen my PPT which I shared with all of you. Have you seen the further PPT? On the basis of what we compare? Sorry Amita, I am not getting your point. What are you asking? On the basis of what we are comparing? What are you asking regarding? We are comparing what? Kindly complete your question. I am not getting it. The protein we loaded on the basis of what we compare. Actually, we are not comparing the proteins. Basically, we are detecting the protein. We have run in the first valve or in the first column, we have placed the ladder of 100 base pair or 500 base pair, or actually we know the size of the base pairs at that ladder, or how much molecular weight the the ladder is carrying like 12 kilodalton or 72 kilodalton. So when we run, if the let me clarify you. One. Yes, yes, I will show you. I will draw here. So suppose this is a gel. We have different size of the, we have run the ladder. So suppose my band is here and we have to compare it with standard proteins or the protein which is required. Suppose the standard protein is of 70 kilodalton. Suppose it. So we will start from here, like this is 15 kilodalton, 50. Then we have 70 kilodalton, we have 90 kilodalton. So if your band comes here in like here or like here, so these are the required proteins which we need further. So standard protein is the criteria on which we are detecting the proteins. In your research purpose or in your work, you have some specific type of protein on which you are working. So standard protein is the one which is your desired result, which you want. We are wanting the protein of 17 kilodalton weight, or we are wanting of 150 kilodalton. So as we move upside, you can say here the protein molecule is of 100 kilodalton. So according to it, we can separate the proteins. So it's a separation of proteins on the basis of their, it is the separation of proteins on the basis of their molecular weight. So on the basis of their molecular, if you can see in this diagram also, the like here, this is, we know the size of these bands. So if the band, like it is a 50 kilo base pair suppose, or 50 kilodalton or 100 kilodalton. So if it comes in between 15 and 100, so its molecular weight may be 70. So by it we will detect the size of the protein. Similarly here, so this is the larger one, so it will come between 100 and 150 kilodalton. So by the help of this, we can get the desired protein which we need for our further work or our further research. So I hope you will get my point, what I want to say. So basically the SDS is generally used for the detection of the protein, the how much size or the separation is generally on the basis of the molecular weight. So are you getting my point? So kindly reply to me. Are you understand what we have done today? This technique is very much important in your ambiguity lecture series. Either you have done southern blotting, northern blotting, you have done anything, but SDS page is very much important. So you have to revise it once again. If you have any query, we will also take it tomorrow. If you may have any queries, so you have to read it thoroughly. So kindly reply me. Are you getting my point, what I want to say? Are you getting it? Do you know the difference between stacking gel and separating gel? Did you know the functioning of SDS? Did you know the functioning of beta-mercaptoethanol or the concentration of your buffer, or how is it work? Today we also done a one demo. I have shown you how SDS page is operating. So do you get it or not? So we have Neha here, Jagruti here, Amita is here, Rahul is here, Juveria is here, Pooja is here. So I hope you all understand it. Tomorrow we will start with your, another topic. I think I will start protein separation. Protein separation is there only. I will start with another. Let me check 1 minute. One minute please. We have another protein purification is there. So we have some another little bit part of biochemistry. So I think we will cover it tomorrow because it's very easy and we will cover it. Yes, you will definitely get the PPT, students. I will provide it to you. No issues regarding it. Don't worry regarding it. Okay, I will share. So maybe tomorrow we will start with a light topic, maybe biochemistry topic like this protein purification, separation and quantification. Maybe we will start it. Have you done microscopy? Kindly let me know because it is also very little and small part. Have you done microscopy, students? बच्चे आप लोगों ने माइक्रोस्कोपी की थी थोड़ी बहुत के नहीं की थी? अमिता, पूजा, आप लोगों ने की थी बच्चे माइक्रोस्कोपी? साक्षी, आप लोगों ने की थी बच्चे? अगर की भी थी तो हम दोबारा करेंगे. आई एम जस्ट आस्किंग कि आपने थोड़ा बहुत पढ़ा है उसके बारे में कि नहीं पढ़ा, बस इतना पूछ रही हूँ. So we will tomorrow maybe we will start protein quantification or separation and purification, or maybe we will start microscopy because both of these are very small and very easy topics. You will easily understand it. If you understand SDS, then not anything is difficult for you. You all are very good students, so I don't think anything will be difficult for you. So please let me know whether you get it today or not. Please let me know. Please. Ma'am, can you show the image of smear on a gel because I cannot get. Actually, what happens in normal separation, Amita, let me clarify. In the normal gel, you will get a straight band. But in case of smear, instead of a single band, you will get little small multiple band on the same location. That is, on a single location you are finding multiple bands. That's a smear, that the concentration is not correct or not regular. So why you are not getting a distinct band like a single straight line? No, you will not find the single straight line, rather you will find the smear. Smear is what? That you are not able to identify the actual point. Are you getting my point? So kindly reply me what we will start tomorrow. कल हम क्या करें? प्रोटीन क्वांटिफिकेशन को स्टार्ट कर लें और we will start with cell microscopy. Kindly let me know. Amita, did you clear my point? What I want to say? Kindly reply me, बच्चे. Kindly reply. हाँ, good. So in the when we have a authentic band, you will get a single band only a single band. And when there is a smear, you will not get a single band, rather you will get small multiple bands, small on the same location, on the same side, but you will get small multiple bands. So kindly reply. हम कल क्या करें बच्चे? बात कीजिए मेरे साथ. Kindly tell me, either we will start with cell microscopy or we will cover the topic which is protein separation, quantification and purification. Kindly let me know, please. Okay, microscopy. Priya is saying microscopy. Protein and then microscopy. Juveria is saying कि ma'am first complete the proteins, then we will go with microscopy. Okay. Kindly share your views first. Yeah, ma'am, but I don't know in exam all are looking like same. Yes. This thing happens in exam. Yes. But don't worry, in exams if you have full confidence, then definitely you will crack that answer. Don't worry, just have little confidence in your own self. This is the only thing. Because in exams we just get aware that now today I have exam, so we will get sometime get little bit you know demotivate, or a little bit fear is in all of us. So the basic thing is that you understand the thing. That is the only point. Your concept should be clear. Otherwise, anything will be okay, but your concept should be clear. Your concept, you should must know the difference between stacking gel and separating gel, the principle of electrophoresis, the principle of running of a gel. Your concept should be clear. Everything will be clear then. So kindly reply me. What should we do tomorrow? So please kindly reply me. So we will. So we will do the things. What with the things which we have to do tomorrow? Okay. Then we will start proteins. We will start proteins then. Someone is messaging me again and again on WhatsApp. So it's okay. You reply me kindly. Kindly reply me. Bache, please reply me. Start with protein. Okay, Amita is saying, ma'am, start with protein. Okay. Okay. Good. Kindly reply me. Kindly reply me. So please, I'm requesting you all again that kindly subscribe my YouTube channel, which is Crash Courses, so that you will aware of the live stream. When I will get live, you will get the notification. So please all the students, kindly subscribe my channel. Ma'am, when we are replying you, but message reads late to you. Yes, I know. This is the problem. I'm getting the message late. But it's okay, students. Just understand my point. If you still have any query, we will discuss it tomorrow. I think that's all for today. That's enough. If you get the SDS page tomorrow, I will ask lots of questions from SDS. So you have to be prepared with it. Okay? I will ask questions. So be prepared with it, and just requesting you to subscribe to my YouTube channel. You will get the notification, and also like my videos also. And may I request you something to all the students, like here I only see your name, like Pooja Yadav, Amita, Rahul is here, then we have Siddhi is here. May I request you to put your photo in your along with your name, like Pooja, so that I will recognize you little bit, like Pooja is here, only Rahul has his pic. So may I allow to ask you what, may I request you to put your photograph here, like Amita is here but no photo is here. So is it possible to all of you or not? It's just a suggestion, it's not a compulsion, it's just a suggestion. It's just a suggestion, bache. It's not a compulsion to any of you, but it is just for the interaction purpose only. So that I will come to know about little more about you, or I will recognize you. Thank you all. So on the last day of this lecture series, when everything will be, our all topics will be, finished, then on the last day, we will do like Google Meet or something to interact with all of you, so that we will meet face to face with each one will meet face to face with each other. So definitely we will do on the last day, either we will do Google Meet. So if you have any type of queries, you may write it at that time. We will deal it personally also. So we will definitely meet on Google Meet or some type of platform where we will able to see each other and we will able to talk with each other. Not only me who is speaking, I want something from you also. So we will definitely do it on the last day. And I don't know when we will meet again or when we will have such type of conversations. So on the last day we will do such type of get together, or you can say such type of meeting where we will, yes, we will meet with each other, we will interact with each other. And on that day I will speak very less and I will ask you to speak. So on the last day we will do this thing. Definitely we will do. And if you will have some type of queries on the 10th day or on the last day, then we will handle it on the last day, or even we will give the chance to the audience or to your friends to speak and answer the question. If you have some questions in your mind, you do not need to be hesitate. You do not need to be shy. You can message me here. You can message me in group also. Because it's your right to ask me a question. It's not any problem. You have to ask me a question. So no issues. We will. You will ask me a question personally also. It's not an issue. You can ask me anything. If you are not clear, you can ask me. No issues at all. Is the connection is okay today? I think today is not any buffering. It happened yesterday only. Now is today the connection is okay? Neha is saying yes. Yes. So I think that's all for today. If you have some question, you can ask. Otherwise, take rest. We will meet tomorrow with new topic, with new energy. Okay, Prachi is saying today no issue, ma'am. Okay, good. Priya is saying yes, ma'am. Today's session is good. So today, if the things working smoothly, if there is not an internet problem, then it's very, we feel very happy to communicate. We feel very happy to study. If there are lots of errors like this network issues or something else, it becomes so disturbing for the teacher and for the students also how to concentrate if such type of hindrance is there. Okay, Jay is saying thank you, ma'am. Sakshi is here. She is saying thank you. No need to say thank you. We all are just friends. We are just friends only. So just this is my request to all of you. Just read the things. So if you have any query, kindly let me know. Sometimes what happens? Sometimes the important point gets missed. We miss it sometimes. So when we will discuss the things again and again, we will focus on the main point. So these things happen. So tomorrow we will meet and definitely I will see your faces, or I will able to see your photos, because today I am not able. Akanksha is here, she has her photo. Priya has also photo, but tomorrow we will see all the students, so it will be good then. So okay students, no need to say thank you. It's not a necessary thing to say. You all are welcome. You always will be welcoming. So that's all for today. We will meet tomorrow. Till then, take care of yourself. We will meet tomorrow. So okay students, should I go now? So I will go now. We will meet tomorrow. Okay. So okay, bye to all of you. Prati, Priya, Juveria, Sakshi, Akanksha, Gayatri, Priya, Revita, Rohit, Mitali, Vishakha. To bye to all of you. Bye. So if any name is left, please don't mind. So we will meet tomorrow. Till then, take care of yourself. Okay. So bye. Bye बच्चे, bye. Take care Juveria, take care बच्चे, bye.